Hello guys, my name is Andres Sava, and today I will be discussing the lab of gummy bears and osmosis. The materials that we will be using today is regular gummy bears, tap water solution, and salt water solution. In this picture, we see the two solutions and the two gummy bears that are placed in the solution. On the left side, we have the gummy bear placed in a tap water solution, a little more clear solution. On the right side, we see the gummy bear placed in a salt water solution with a higher concentration of salt. And we see how it's not that visible, the gummy bear in the cup. Gummy bears are made up of three things: sugar, starch, and gelatin. The gummy bears have a selectively permeable coating, which allow water molecules to diffuse across but inhibit larger molecules to pass through the membrane of gummy bears. Osmosis is a passive process that do not require energy. It involves the movement of molecules from a region of higher concentration to a lower concentration until the concentration becomes equal on either side of the membrane. Osmosis is important in living organisms as it influences the distribution of nutrients and the release of metabolic wastes products such as urea. There are three types of osmotic solutions: isotonic osmotic solution, which is a solution that has the same concentration of solutes both inside and outside the cell; hypotonic osmotic solution, a solution that has a higher solute concentration inside the cell than outside the cell; hypertonic solution, a solution that has a higher solute concentration outside the cell than inside the cell. In this picture, we can see the three types of osmotic solutions. We see in the first picture an isotonic solution where the concentration is the same outside and inside the cell. We see a normal cell right here. We are looking in a red blood cell. In the second picture, we see a hypotonic solution. The cell is placed in a hypotonic solution, and we see that the concentration outside the cell it's lower than inside the cell. So the water from outside the cell tends to move inside the cell. That's why the cell looks a little more diluted and more enlarged than the first picture. And in the third solution, we have a hypertonic solution where a red blood cell is placed in a higher concentration solution than the concentration of inside the cell, and we see uh, water movement of um, red cell moving outside of the red cell, resulting in a more shrink, or I would say the cell becomes a little smaller. There are two types of osmosis. Endosmosis, when a substance is placed in a hypotonic solution, the solvent molecules move inside the cell and the cell becomes a large, swollen, or undergoes deplasmolysis. Exosmosis is when a substance is placed in a hypertonic solution, the solvent molecules move outside the cell and the cell shrinks or undergoes plasmolysis. The results of the experiment is we place both gummy bears in two different solutions, and we let them sit there for about 7 to 8 hours. Um, in the first picture, we see a normal gummy bear. In the second, we see we can compare the two gummy bears, the tap water gummy bear to the normal gummy bear. We see that the tap water gummy bear is a little more uh, bigger than the normal gummy bear, and that is because the tap water solution is a lower concentration solution than the gummy bear, resulting in water from outside going or moving inside the cell or the gummy bear, we can say, which is an example of osmosis. Um, and the third gummy bear we see it's a little smaller than the other two gummy bear, and that is because the salt water is a higher concentration solution, than the inside of the cell or the gummy bear, and it's an example of exoosmosis, which water molecules from the gummy bear move outside the membrane of the gummy bear in order to um, dilute or create an isotonic environment, and that results in shrinkage, or the gummy bear gets smaller and smaller.